Hello everyone, welcome to our YouTube platform Concept and Fact of Life Sciences. Today in this video we are going to discuss about the nitrogen metabolism. Nitrogen, it is 78% on the earth surface, but still we need to fix the elemental nitrogen. Why? On the other hand, the amount of oxygen on the earth crust, it is only 21%. But still this oxygen, it do not require any fixation. And what is the need to fix the elemental nitrogen? There is a triple covalent bond which remain present between the nitrogen atom which forms the nitrogen. This nitrogen, it is able to enter into the plants through the stomata, but it is of no use. Why? Because this nitrogen, it is not having the sufficient energy that can be added onto the keto acid to convert them into the amino acids. So, therefore, we need to convert this elemental nitrogen into the higher grade form or high energetic form of nitrogen. The absorbed form of the nitrogen, it is either the ammonia or the nitrates. If we talk about the energy content, the maximum amount of energy it is remain present within the ammonia, followed by nitrate, nitrite, and the least amount of energy remain present within the nitrogen. So there is a need to convert this elemental nitrogen into the usable form, that is the ammonia, which can be added onto the keto acid to convert them into the amino acid. So therefore there is a need for the conversion of this elemental nitrogen into the usable form. Now any process which converts the elemental nitrogen into its usable form, that is the absorbed form, nitrate or the ammonia, it is known as the nitrogen fixation simply. Now this nitrogen fixation, it is of two types. One is the industrial nitrogen fixation while the other is the natural nitrogen fixation. Industrial nitrogen fixation, it includes the Haber process. Haber process, there is a conversion of nitrogen and hydrogen under the high atmospheric pressure and high temperature into the fixed or absorbed form, that is the ammonia. And this industrial process, it contributes around 80 into 10 to the power 9 kilogram of fixed nitrogen per year. On the other hand, the natural nitrogen fixation, it contributes 180 into 10 to the power 9 kilogram of fixed nitrogen per year. Now this natural nitrogen fixation, it is further divided into two types. One is the photochemical reaction while other is known as the biological nitrogen fixation. Photochemical reaction, there is a conversion of nitric oxide in the presence of ozone and UV radiation into the nitrates, and this photochemical reaction, it contributes only 19 into 10 to the power 9 kilogram of fixed nitrogen per year. Now the major contributor, it is the biological nitrogen fixation. Here the organisms convert the elemental nitrogen into the usable form. And under the biological nitrogen fixation, these organisms can be divided into two types. One is known as the free-living nitrogen fixers, while the other is known as the symbiotic nitrogen fixers. The free-living nitrogen fixers, they can be further categorized into aquatic. These includes the cyanobacteria, nostoc and anabina. There is aerobic bacteria in the form of azotobacter as well as the azospirillum. There are facultative anaerobes like Klebsiella, which carry out the nitrogen fixation under the anaerobic condition. There is anaerobic bacteria in the form of Clostridium as well as some archaebacteria in the form of Methanococcus. There is photosynthetic aerobes, which includes the purple sulfur bacteria as well as some green sulfur bacteria. On the other hand, there are some symbiotic nitrogen fixers in the form of rhizobia. Rhizobia, they forms the symbiotic association with legumes as well as some non-leguminous plant, for example, the parasponia. While the other category, it includes the Frankia, which forms the symbiotic association with non-leguminous plant, for example, the Casuarina. So these are the nitrogen fixers, or we can say biological nitrogen fixers. Now, all of these organisms, to convert the elemental nitrogen into the usable form, they have an enzyme which is known as nitrogenase enzyme complex. And this enzyme, through this enzyme, they are able to convert this elemental nitrogen into the fixed form. Here we are converting a less energetic form into a more energetic form. Therefore, we require or we need to invest some amount of energy, that is the 16 ATP. During this process or the reaction which is catalyzed by the nitrogenase enzyme complex, we require 8 H plus ion and 8 electron, which comes from the 8 ferredoxin molecules. Now this nitrogenase enzyme complex, it is made up of two complexes, or it is divided into two complexes. One is known as dinitrogen reductase complex, while the other is known as dinitrogenase complex. The dinitrogen reductase complex, it is made up of two subunits. It is also known as Fe4S4 complex, while the dinitrogenase, it is made up of four subunits and it is also known as FeSMO complex. During the reaction which is catalyzed by the nitrogenase enzyme complex, the electron donor is ferredoxin. When there is a conversion of reduced ferredoxin into the oxidized ferredoxin, they will give their electron to the Fe4S4 complex, and this oxidized complex will convert into the reduced one. Whenever there is a oxidation of this Fe4S4, there is a reduction of another complex which is known as FeSMO complex. And when this FeSMO will oxidize, it will transfer the electrons to the nitrogen. So there is a conversion of this nitrogen into the ammonia. Now here these 8 H plus ions, these comes from the bacterial cytoplasm. The byproduct of this nitrogen fixation, it is the hydrogen. There is a production of hydrogen as a byproduct during the nitrogen fixation. Now, for a pair of electron, for two electrons to be transported from dinitrogen reductase enzyme complex to the dinitrogenase enzyme complex, we require the investment of 4 ATP. Now, for the transportation of 8 electron, there is a requirement of 16 ATP. So, for the transportation of 2 electron, we require 4. So, for transportation of 8 electron, we require 16 ATP of energy, जो अपन ने यहाँ पे भी बताया हुआ है. Now, out of this 16 ATP, we can say for the production of ammonia, we require only 6 electrons. 6 electron means we require only 12 ATP energy, while remaining 4 ATP energy or for the transportation of these 2 electrons, which is required for the synthesization of byproduct, it is total waste. But some modern organism or some modern bacteria, they have both nitrogenase enzyme complex as well as the hydrogenase. Now this hydrogenase, it is able to convert this hydrogen, that is the byproduct of nitrogen fixation, by the process that is called as reversible oxidation into the H plus ions. Now these H plus ions, they can be utilized over here during the next round of nitrogen fixation, we can say. Now, the nitrogenase enzyme complex, it is strongly inhibited or it is susceptible for the oxygen. Now, upon exposure to the oxygen, this dinitrogen reductase complex, it will, we can say, inhibited within the seconds, while upon exposure to the oxygen, the nitrogenase enzyme complex, it will inhibited within the minutes. So कुछ ही सेकंड ऑक्सीजन के एक्सपोजर के बाद में, कुछ ही सेकंड बाद में ये कॉम्प्लेक्स काम करना बंद कर देगा और कुछ मिनट्स बाद में ये कॉम्प्लेक्स काम करना बंद कर देगा. सो सारे जो आपके पार्ट्स हैं, कॉम्प्लेक्सेस हैं नाइट्रोजनेस एंजाइम कॉम्प्लेक्स के, वो बहुत ज्यादा ससेप्टेबल हैं किसके लिए? ऑक्सीजन के लिए. सो द बैक्टीरिया दे हैव टू मेंटेन और दे have to have some special conditions where they can carry out the nitrogen fixation under the aerobic condition, or we can say they have to have certain adaptation by which they can carry out the nitrogen fixation under the aerobic condition. Now, for cyanobacteria, these cyanobacteria, these are the algae, they have filamentous plant body. Now, some of the vegetative cells, these are the photosynthetic bacteria, and during the photosynthesis, as we all know, there is a production of oxygen. Now, some of the cells of filament, they stop the photosynthesis, they secrete a thick wall around them, and these cells are now called as the heterocyst. So, there is no photosynthesis. So, these cells, they are able to carry out the nitrogen fixation because the oxygen is not there, and they will provide the fixed form of nitrogen to the nearby cells and will take up the nutrients, that is the product of photosynthesis, from the nearby cells. So it is a symbiotic association within the bacteria. Second is the free living nitrogen fixers, that is the azotobacter. Under the nitrogen deficient condition, when they require to fix the elemental nitrogen, they increases their respiration rate. It is one of the strategies. Klebsiella, it is facultative anaerobe, so they will carry out the nitrogen fixation under the anaerobic condition. So they only fix the nitrogen under the anaerobic condition. So there is no, we can say, there is no effect of the oxygen onto the nitrogenase enzyme complex because they are living into the anaerobic condition. Now, next category is the purple sulfur bacteria. Now, during the process of photosynthesis in case of plant, the electron donor is H2O. But in case of sulfur bacteria, the electron donor is H2S. So, the product of this photosynthesis is sulfur, not the oxygen. So, this sulfur, it is not going to affect the nitrogenase enzyme complex, so they can carry out the nitrogen fixation. In symbiotic bacteria, the plant helps the bacteria to carry out the nitrogen fixation. For this, the plant releases leg hemoglobin in the nodules, and this leg hemoglobin, it is having more affinity for the oxygen as compared to the hemoglobin. So, whichever the oxygen which is produced in the nodules, it will be taken up by the leg hemoglobin. Now, first we discuss about the symbiotic nitrogen fixation, which will be followed by the free living nitrogen fixation. So if we talk about the symbiotic nitrogen fixation, it depends upon the signaling between the plant and the bacterium. Now the plant, it is the plant root, it is the plant root here. These roots, they releases certain chemotactic signals in the form of flavonoids and isoflavonoids for the root nodulating bacteria which remain present in the soil. So the roots, they releases certain chemotactic signals in the rhizosphere. Rhizosphere is the soil which surrounds the root system. So in the rhizosphere, there is a release of flavonoid and isoflavonoid which can be sensed or received by the root nodulating bacteria which remain present in the soil. In this root nodulating bacteria, so these are the bacteria. So they are prokaryotic system, and in prokaryotes there is a polycistronic type of transcription. Means many genes, they shares a common promoter. Now here a gene complex which is having node D gene, node A, B, C, E, F, L gene. The node D gene, which remain present in these root nodulating bacteria, it is having a constitutive expression. Means the gene which always express, it is known as constitutive expression. So due to the constitutive expression of node D gene, there is a production of node D protein. That is the node protein. This node protein, when it receives this flavonoids, they are now able to recognize the promoter sequence and they are going to bind with the promoter sequence after binding with the isoflavonoids and flavonoids. After binding to the promoter sequence, this promoter sequence, it will be recognized by the sigma 54 factor of bacteria. Then there is a binding of RNA polymerase and there is an expression of these gene, which is ABCEFN. Now these ABC genes, they code for the structural component of node factor. The node factor, it is basically a lipochitin oligosaccharide. Now structure component, it will be coded by the node ABC gene, while the E and F gene, they will determines the chain length and the unsaturation in this node factor, while the L, it provides the specificity to the node factor, which is specific. Now, combinedly, the node factor, it is a lipochitin oligosaccharide which will be produced in response to the flavonoid and isoflavonoid which is produced by the or which is released by the root hairs or roots. Now, the root nodulating bacteria, they will, due to the chemotactic signal, they will start moving towards the root hair. The root hair, they are having a receptor which are called as lectin receptors. These lectin receptors, these are basically the receptors of sugar. So they receive the sugar, that is the lipochitin oligosaccharide. There is presence of sugar. So they receives the node factor, and after receiving the node factor, there is a curling of root hair. There is a curling of root hair, which is mentioned over here. And there is a colonization of these root nodulating bacteria in this curled region. After this, due to the fusion of Golgi-derived vesicle, there is a formation of infection thread in the root hair. The bacteria enters into the root hair through this infection thread and reaches towards the cortical region. Thereafter, there is a fusion of endomembranous system. There is a fusion of endomembranous system, that is a Golgi and endoplasmic reticulum, which moves this thread from epidermal regions to the cortical region of plant root. Now, within the cortical regions, there is a formation of vesicles type of structure. These vesicles, finally, they will get detached from the infection thread. Now, the membrane which remain present around the bacteria, it is called as peribacteroid membrane and it is of plant origin. While the bacterial cells which remain present within the peribacteroid membrane, they are called as bacteroids. So after this, there is an expression of nodulation genes in case of plant. अभी तक जो काम कर रहे थे वो बैक्टीरियल जीन्स काम कर रहे थे, अब कुछ प्लांट्स के नोडुलेशन जीन एक्सप्रेस होते हैं. देयर इज एक्सप्रेशन ऑफ नोडुलेशन जीन्स ऑफ प्लांट व्हिच produce huge amount of auxin and cytokinin, due to which there is a rapid cell division that leads to the formation of nodule-like structures. Now, another type of nodulation gene, it will express, lead to the formation of leg hemoglobin. As we already stated, here the plant releases leg hemoglobin, which takes up the extra amount of oxygen and allows the bacteria to carry out the nitrogen fixation. Now, there is a production of lag hemoglobin. After the production of lag hemoglobin, the node gene, it stops its expression. So now the node gene complex, it stops its expression. So there is no production of node factor now. At the same time, there is an expression of, or we can say this lack hemoglobin, it induced the expression of another type of genes in bacteria, which is called as Nif genes. Now these Nif genes, these Nod and Nif genes, they are called as symbiotic genes. Now these Nif genes, they code for the nitrogenase enzyme complex, which is the crucial enzyme for the conversion of elemental nitrogen into the usable form. Now, there is a conversion of elemental nitrogen into the ammonia within the cortex of plant. Now, within the pH of pH environment under the cortical cell, it is like, we can say, alkalinic, or it is around 7. So at the physiological pH, it will remain in the form of ammonium ion. There is a presence of vascular tissue, that is the xylem nearby, which can carry out the transportation of this fixed form of nitrogen to the aerial parts of the plant. But the xylem, it is not able to transport the nitrogen in the form of ammonia. We have to convert this ammonia into the suitable transportation form. And here there are two suitable transportation forms. One is called as the amide forms, which includes the glutamine and asparagines, while the other includes the urease form. The transportation form, that is the amide, it is generally carried out by the plants in the temperate region, while the plants of tropics or the tropical regions, the transportation form of ammonia is urease. Now, the amides, there is an conversion of glutamate by the addition of ammonia into the glutamine in the presence of glutamine synthetase. It is a synthetase, so it utilizes the energy in the form of ATP. ATP will convert into the AMP. So the glutamate, which is a acidic amino acid, it will be converted into its amide form, that is the glutamine, by the addition of ammonia in the presence of enzyme GS. Now this glutamine, it will be added onto the alpha ketoglutarate, which is a keto acid. So there is a one amino acid and other is the keto acid. Now this glutamine, it will release the ammonia. This ammonia, it will be taken up by the alpha ketoglutarate and will form one unit of glutamate. While after releasing the ammonia, the glutamine, it will be converted into the another glutamate molecule. So there is a production of two glutamate. One of the glutamate, it will be added on the oxaloacetic acid, will be converted into the aspartate, and the aspartate in the presence of enzyme asparagine synthetase will be converted into the aspargine. So this glutamate, it will be utilized in the next cycle. So this is the transportation of ammonia in the form of amides. But if we talk about the ureides, if the ammonia is taken up by the uric acid, it will be converted into the allantoin. This reaction, it occurs within the peroxisomes. It will occur in the peroxisome. Now, this allantoin, it will be hydrolyzed. There is a hydrolysis of this allantoin into the allantoic acid, which occurs within the endoplasmic reticulum, that can be transported by xylem to the aerial part of the plant. So it is the suitable transportation forms of ammonia. Now, another category, that is the non-symbiotic nitrogen fixation. Here, there is a conversion of this elemental nitrogen again into the ammonia. There is a requirement of 8 H plus ion and 8 electron, and there is a production of byproduct, which is the hydrogen, and it is the free living nitrogen fixation. So it occurs within the soil, the pH of the soil. The ammonia will also remain in the form of ammonium ion. This ammonium, it is highly volatile and it cannot be retained in the soil for prolonged period of time. But if the free living bacteria, in the form of nitrosomonas and nitrobacter are there, it will be converted into the absorbed form or in the form which can be retained in the soil for prolonged period of time. So ammonium, if there is a presence of nitrosomonas, it will be converted into the nitrite. So there is a conversion into the nitrite through the nitrosomonas. There is another class of bacteria which are called as nitrobacter. They will convert the nitrite into the nitrate. There is a conversion of nitrite into the nitrate, and these bacteria, they are called as the nitrifying bacteria. These are chemoautotrophs. We are here converting a higher energetic form into the less energetic form. So, some amount of energy which is released, it will be utilized by the bacteria for their own metabolism. The bacteria, they are also doing it for their own purpose. Although they are helping the rhizosphere to retain the fixed form of the nitrogen for prolonged period of time, but at the same time, they are doing this for their own purpose. They are selfish. They are doing this because they are converting the higher amount of energy, higher grade form of energy into the lower grade form of energy. There is a release of energy, and they are known as the chemoautotrophs. They utilize that energy in for their own metabolism. Now, there is a presence of nitrate in the soil. Now, there is a disadvantage of conversion of this ammonium into the nitrate. First is that the silica soil particles, they are also negatively charged. The nitrates, they are also negatively charged. So there is no ionic type of interactions are there. So this nitrate, it can be washed away with the surface runoff. Next thing, the plant absorbs the nitrogen in the form of nitrate. But within the plant, these nitrates, they have to be converted back into the ammonia, so that can be added onto the keto acid to form the amino acid through the ammonia assimilation. So there is two disadvantage of conversion of this ammonia into the nitrate. Now, so the available form in the soil, it is nitrate. The root here, plant root here, they have proton pump. This proton pump, it utilizes the energy in the form of ATP and will transport the H plus ion against the concentration gradient. Now there is another kind of transporter which is called as proton nitrate symporter. This proton gradient, it is utilized to transport the nitrate within the plant. Now the nitrate, it remains present within the plant. Now within the plant, they have to be converted back into the nitrite by the help of enzyme which is known as nitrate reductase. There is another enzyme which converts this nitrite into the ammonia, which is known as nitrite reductase. So there is a ammonia assimilation. यही बात अपन यहाँ कर रहे थे. There is a disadvantage of conversion of this ammonia into the nitrate, क्योंकि प्लांट के अंदर जाने के बाद उसको वापस अमोनिया में कन्वर्ट करना है, and that process is called as ammonia assimilation. This process is called as nitrification. So this is the non-symbiotic नाइट्रोजन फिक्सेशन. बात करते हैं दोनों एंजाइम्स के बारे में जो इंपॉर्टेंट है, नाइट्रेट रिडक्टेज और नाइट्राइट रिडक्टेज के बारे में. नाउ ammonia assimilation, the first enzyme here, it is the nitrate reductase. The nitrate, it is within the plant. There is an enzyme which is called as nitrate reductase, which requires 2 hydrogen and 2 electron, which comes from the reduced NADPH. It will get oxidized, carry out the reduction of nitrate into the nitrite. This nitrate reductase, it is an dimeric enzyme which is made up of 2 subunits, and it is a cytosolic enzyme. It remains present within the cytoplasm of plant. Okay. Now it is a dimeric enzyme. It is first unit and it is the second unit, and these units, they contain 3 prosthetic groups. Now the enzyme, it requires certain cofactor for their functioning, and these cofactor, they may be the coenzyme or the prosthetic group, and this nitrate reductase, it requires 3 prosthetic group in the form of FAD, heme and molybdenum. It is the only enzyme in the plant, that is the nitrate reductase, which requires molybdenum as a cofactor. Now there is a electron donor, which is the NADPH. Now this transfer their electron to the FAD. FAD will convert to the FADH. When it will get oxidized, it will transfer the electron to the heme. The heme will transfer the electron to the molybdenum, and finally the electron will terminate into the nitrate, and there is a conversion of this nitrate into the nitrite, and there is a release of water molecule. So this is the functioning of nitrate. This is the functioning and structure of nitrate reductase, which is a cytosolic and dimeric enzyme. Now next is the nitrite reductase. This nitrite reductase, it converts the nitrite into the ammonia. The second step of ammonia assimilation, it requires 6 electron and 8 H plus ion for the conversion of this nitrite into the ammonia, and this 6 electron, they comes from 6 ferredoxin molecules. Now this ammonia which is produced over here, it is also toxic for the plant. It can act as the uncoupler. It can act as the uncoupler, which can disturb the membrane gradient, or the gradient across the membrane. So there is a need for the conversion of this ammonia into the amino acids, and for this we have a pathway which is known as GSGOGAT pathway. Okay, there is a glutamine synthase, glutamate oxoglutarate amino transferase reactions for the conversion of this ammonia into the amino acids. So first of all, there is a conversion of this ammonia, or addition of this ammonia onto the acidic amino acid, which is known as glutamate. There is a requirement of energy because it is a synthetase type of enzyme. It will be converted into the glutamine. This glutamine, which is the amide of this glutamate, it will be added on the keto, alpha ketoglutarate, which is a keto acid, 1NH3. It will be released and it will be taken up by the alpha ketoglutarate, as we already discussed. There is a formation of 2 glutamate molecule, one by releasing the ammonia and other by taking up the ammonia which is released from the conversion of glutamine into the glutamate. The enzyme which functions over here is the glutamate synthase. So there is a formation of 2 glutamate, and out of 2 glutamate, one, it will be ready to accept the another ammonia molecule, and in this way there is a conversion of this ammonia into the amino acids. Now this nitrate reductase, we can say this nitrate which is produced or which remain present within the plant, it will be converted into the nitrite, and this nitrite, we can say, it will be converted into the ammonia by the another group of enzyme which is called as nitrite reductase. This process is called as the ammonia assimilation. Now this nitrate reductase, it is a cytosolic enzyme, while this nitrite reductase, it remains present within the plastids. So this is all about the nitrogen assimilation in plants.